horrible and crippling disease. Okay, right, here we go. Let's start presenting here. I want to talk about, continue to talk about this mass of organisms called the protista, which again don't form one single kingdom. Current thinking divides up the eukaryotes into 5 separate kingdom-level taxa. We talked about one of them on Monday. That's the amoebozoa, the amoebas. The second group I'm going to hit is affectionately known as SAR. It doesn't really have a proper, Latin-style name as yet. And the 3 groups that make it up are Straminopiles, Alveolata, and Rhizaria. The Alveolata are the ones that we're going to focus on because they contain a subgroup called the Apicomplexa, which includes the gregarines that we're looking for and that 8 of you have seen in lab. The Apicomplexa also includes things like coccidia, which is very important veterinarianally, whatever that word is, and malaria is another big one. So we're going to start by talking about SAR, mostly focusing on apicomplexans, which are a subgroup of the alveolates. And then excavata is the other group that includes an awful lot of protist parasites. And then the other 2 are Opisthocanta, which has animals, fungi, and some other single-celled critters, and Archoplastida, which would be plants, red algae, and green algae, and a few others. SAR, we only think it exists because of similarities in DNA sequences. I'm assuming that everybody has had some exposure to cladistics before, and knows that the way we try to classify living things is by our best estimate of their common ancestry. And one important way that we can try to work out common ancestry is for looking for patterns of similarities and differences in DNA. For SAR, the groups that make up SAR are so different from each other that there's no obvious features of structure that they all share. There's nothing I can point to in a cell and say, this is a typical feature of SAR. They're so different from each other that similarities in A's, T's, G's, and C's are all we really have to go on. Members of SAR include brown algae, such as kelps, golden algae, like the thing you see on the left. That's a freshwater alga called, I think that's Chrysobryon. You can see little yellow spots in the cells. Those are chloroplasts. And then a group of very prominent protists in the fossil record because they make skeletons. In the center, that's a radiolarian, and on the right, that's a foraminiferan. Those of you who are victims of my invertebrate zoology course might remember those. But none of those are parasites. We won't be talking about them further. SAR also includes a number of important plant parasites. We won't be talking much about those. This is animal parasitology. But on the top, you have an organism called Phytophthora. It used to be classified as a fungus, and superficially it looks like one, but the similarities are due to convergent evolution. It's not really a fungus. Phytophthora actually causes quite a lot of plant diseases. One in particular, Phytophthora infestans, might be the reason why some of you are here, because Phytophthora infestans is what attacked the Irish potato crop starting in 1845 and caused 5 years of famine. Something like 1 million Irish people starved and roughly another 1 million emigrated to this country. So if you've got Irish ancestry, Your ancestors might be here because of Phytophthora infestans, which wiped out the crop that was supporting most of the Irish population. There's lots of other plant blights that are caused by organisms related to Phytophthora in a group called the oomycetes, common name the water molds. Which used to be classified as fungi. We now know that they aren't. They're actually often this SAR group, specifically in the subgroup Stramenophiles. They also include this thing called Plasmodiaphora. That's seen at the bottom. That's not an oomycete. It causes something called cabbage clubroot disease. Here, I just heard something go ding dong, so. Okay, somebody joining. Okay, I've just seen the question asked if anybody is having audio issues. If you are, let me know, but somebody else responded that things seem to be going okay. So I'm sorry if anybody's having them. I can't really tell from here. Anyway, again, Plasmodiaphora also used to be classed as a fungus. It's certainly a problem if you're raising cabbages, and it can attack some other vegetable crops as well. These belong in this SAR group, and they're parasitic, but they don't bother animals. There are some water molds, this oomycete group, that can affect animals. This is an aquarium fish that's attacked by saprolignia. Saprolignia very often is just a decay organism. It's a decomposer. Sometimes it can attack live fish, usually as a secondary infection, meaning the fish gets wounded and opportunistic bacteria and protists, including saprolignia, can enter the wound and start growing. As far as I know, I don't think these normally pose a health risk for healthy fish, although I think I did read that sometimes you do see it growing on fish that didn't get wounded. So I don't know much more about it than that, but sometimes this will be an opportunistic parasite on fish. There's another one called Pythium that is pretty rare, but it can cause very severe lesions. You know, lesion is medical talk for boo-boos on dogs, cats, horses, and very occasionally people. It's not very common, but it's pretty bad when it does hit. When it affects horses, one name that you'll see for it is swamp cancer, because it makes these tumor-like swellings under the horse's skin, and because it tends to flourish in wet environments. If you've been riding your horse through a swamp, it might be more likely to get it. So those of you interested in vet school might see this again, but it doesn't seem to be very common. One more group within SAR that I'll talk about very briefly is called the Opalinia. Now, one of the things I love about biology is that life has this wonderful way of colonizing any niche that it can. So naturally, there has to be something living in frog rectums, because why not, right? If we have things living in cockroach guts, then there's got to be somebody making a living inside frog rectums, because it just wouldn't make any sense if they didn't. And this is a large protist called Opalinia. I have not had the pleasure of seeing them live. They used to be commonly demonstrated in parasitology classes, but the standard way of getting them is killing a frog and cutting open its rectum. And that might have been okay back before amphibian populations started declining. But in this day and age, A, it takes too many hoops to jump through to kill vertebrates in your class, and B, I don't particularly want to go out and kill frogs if I don't have to. So you may never get to see these guys. I'm told they are called opalinia because you don't see it in this picture, but they shimmer like opals if you see them live. They're covered with many flagella, but they're not actually ciliates. They are more closely related to the Straminopiles group, which includes the oomycetes, but it also includes brown algae, golden algae, and things like that. And as far as I know, they don't cause a great deal of damage to the frog. They just swim around in the frog rectum because somebody has to. I mean, I suppose, you know, they got their degree from Arkansas Tech or something like that, and the only jobs they could get involved living in frog rectums. I hear that happens a lot. They live in frog rectums as multinucleate adults, and it's not super obvious on this slide, but if you look carefully, you can see a sizable number of darker blue round spots in this opalinia. That's nuclei. So they live as single cells with multiple nuclei, and then they divide without duplicating their nuclei. Each nucleus just ends up in a division product. So just remember, nuclear division and cytoplasmic division don't have to happen at the same time. And when they divide without duplicating their nuclei, they end up forming little cysts, each of which has a single nucleus. The cysts come out in feces, and then they get eaten by tadpoles. Within the tadpole, they'll undergo a sexual stage and then develop into new adults. And that's all the opalinia I've got time for. There's one more in SAR called blastocystis, which used to be considered a yeast. It's only fairly recently that studies of its DNA revealed that it's not a yeast, which are fungi. What it is, in fact, is a protist in this SAR group. And it is said to be the most common protist living inside humans, with an estimated 23% of us in the USA infected with it. The last paper that I looked at talking about blastocystis, there was still debate, at least there was as of a couple of years ago, as to whether this is a parasite at all, because most people that have this don't have any obvious symptoms. So most of the time, this seems to be purely a commensal. You know, you might have it and never know and never need to know. It might never be detected unless they went looking for it. If you had to have a stool sample looked at or something like that. There have been some strains that have been linked to IBS, irritable bowel syndrome, or to similar symptoms, gas, diarrhea, weakness, sometimes skin problems, things like that. So some strains of this might qualify as parasites. Last I looked, it still wasn't completely known. I think there's a lot about blastocystis that nobody's bothered to look at. So this may turn out to be an important human parasite. But again, most people that have it never seem to develop any symptoms at all. So maybe it's just one particular subtype or subspecies within the genus that might cause symptoms. But again, this is something that needs to be studied further. We do know that it's transmitted by the fecal-oral route. Remember that word, we use it a lot in here. In the body, there's a fairly complicated life cycle. The cysts that pass from one person to another, by a person consuming something that's been contaminated with feces from an infected person, that person will swallow cysts. And in the body, the cysts release a form called a vacuolar form, which I think is what you were looking at on the previous slide. They might reproduce by multiple rounds of asexual division, which is called schizogony, and that's a word you're going to see again. or they might turn into amoeba forms that eventually form new cysts. So again, kind of a mystery there as to what they're doing. Okay, that's all I'm going to say about the S and the R in SAR, the Stromenopiles and the Rhizaria. The vast majority of those aren't parasitic at all. The A in R stands for the alveolata, and all three of these have some parasitic members. Dinoflagellates we'll talk about later. Ciliophora is the ciliates. If you saw paramecium back in your intro classes, you've seen ciliates. We see several other species in invertebrate zoology, and they're abundant in water samples. So if you've ever looked at pond water under a microscope, you have probably seen ciliates. They're very common in the oceans as well, and 8 of you have seen a ciliate that lives as a commensal in cockroach guts. That's that kind of pear-shaped, slowly spiraling thing that we've seen called Nyctothyrus. Those of you that haven't seen it will, either this Thursday or if you can make it up this week, that'd be great. Apicomplexa is the one that includes most of the parasites. That's the one that I'm kind of working up towards, because that includes gregarines, coccidians, a group called the pyroplasms, and a group called Hemosporidia, which is the malaria parasites. These 3 are grouped together as a clade because of similarities in DNA, but they've also got a structural feature in common. The cells have outer membranes, like all cells do. Just under the outer membrane, they've got another set of membranes which form flattened sacs. So there's a set of flattened membrane sacs, almost like a quilt, underneath the external membrane, and that's called the alveolar membrane system. And there's what it looks like. On the left, you've got some dinoflagellates. On the right, you've got a ciliate, paramecium. In the middle, that's what a malaria parasite looks like, Plasmodium. These have been stained so that the nuclei are staining blue. And proteins that are unique to the alveolar sacs, called alveolin proteins, are staining green. And you can see that all 3 of these have this green layer outlining the outside. That's where the alveoli are located, just inside the outer membrane itself. And again, I find a metaphor of a quilt is a pretty good one, because those membrane sacs are very flattened, and at least in ciliates, I guess in all of them, they fit together very tightly. In dinoflagellates and at least some ciliates, they're involved in rigidity of the cell, helping it hold its shape. Okay, so within that alveolata group, the big subgroup we're going to focus on is the Apicomplexa. This will probably take us about a week to get through. And they're called Apicomplexa because they all have organelles at the anterior end of the cell called the apical complex. That's what's used to attach to a host cell and penetrate, sometimes partially, and in many cases completely, into the host cell. When you have a malaria infection, for example, one of the reasons why it's hard to treat is that the malaria parasite spends an awful lot of its life cycle hunkered down inside your red blood cells and or your liver and spleen cells. It's an intracellular parasite, as we say. and it's able to penetrate into host cells using this structure called the apical complex. Most apicomplexans also contain a plastid. Now, I assume you know about chloroplasts. Those are the little green dots inside plant leaves. I don't know if you get to see these in 1440, but in 1400, we get everybody to look at live chloroplasts in the leaves of a water plant, and I'm assuming you've seen these before. The plastids of Apicomplexa look like chloroplasts. The problem is they're not photosynthetic. Apicomplexans would spend almost all of their life cycle in complete darkness, right? It's dark inside your liver. It's dark in most of your blood vessels, except maybe the surface capillaries of your skin. So the plastid is not used for photosynthesis, and exactly what it does, it's got some interesting biochemical roles, and it seems to be evidence for common ancestry with dinoflagellates, which usually do have a plastid that they use for photosynthesis, but we'll talk about them later. There's never any cilia or flagella. Some stages of the life cycle may not move at all. Those that can move, they kind of crawl or glide, meaning they move, but you can't quite tell how they're doing it. At least a couple of you have gotten to see this. They're all parasitic in animals. In addition to malaria, human diseases include toxoplasmosis and cryptosporidiosis. Those are not always particularly bad. You can have toxoplasma infection and never know it, but they're particularly dangerous for people with compromised immune systems. They used to be very common in people with full-blown AIDS. I guess they still are. There are risks if you've had an organ transplant or otherwise are immunocompromised. Some economically important ones that vet people might encounter. Coccidiosis is a real problem in poultry, chickens, the Arkansas state bird. Dogs and cats can get it as well. Coccidiosis is a leading cause of severe diarrhea in puppies. Another one called babesiosis causes red water fever in cattle and can occasionally infect humans as well. And the life cycles start getting a little complicated. And one of the things that you are going to have to know backwards, forwards, over, under, sideways, down is the life cycle of the malaria parasites. There'll be handouts for that, and we'll get to that very soon. The main parasitic stage is haploid. The stages that 8 of you have seen under the microscope, and the rest of you will soon, those stages were all haploid. The haploid stages pair up and they form cysts called gametocysts, and inside those sexual reproduction takes place. Many species, although not the gregarines that we're looking at this week, many species alternate sexual and asexual reproduction. They'll reproduce sexually and alternate that with rounds of asexual reproduction. In malaria, the sexual stages are all in the mosquito. They only reproduce asexually in humans, but we'll get to that soon enough. And that's what the apical complex looks like. You have a couple of rings of microtubules called the apical rings, sensible name for them. Well, you've got the apical rings and then the polar rings. And between the two of those, you have a structure made of spiraling microtubules called the conoid. And then radiating for that, you have microtubules that run along the body axis just under those alveolar sacs called subpellicular microtubules. They're probably involved in locomotion. It's probably these that contract in some way to enable apicomplexans to glide along and sometimes change their shape a bit. So in this picture, you can see the apical ring and the polar ring. Between the two of them, you have that spiraling thing, that's the conoid. Right where it says inner membrane complex, that's the alveolar membranes. That's that flat membrane sac underneath the outer membrane, which is labeled the plasma lemma here. And then radiating out from the polar ring, you have those subpellicular microtubules that might run all the way down the length of the cell. So this is the nose cone that's involved in attachment to hosts. And that's a picture of it. That's a transmission electron micrograph prepared by a process called negative staining. And the part up at the upper right that looks almost like it's checked or clad, that's the conoid. That's those spiraling microtubules. You can see the apical ring and the polar ring above and below the conoid, and then you can see those long longitudinal microtubules extending down. So this is what it looks like in Toxoplasma, a parasite that alternates between rats and cats and can sometimes affect humans as well. This is a cross-section through Toxoplasma inside a host cell. You can see the conoid is labeled C over there on the far right. Extending back from it, you have some very slender fibers called micronemes, labeled M, and some long bag-like things that run from the conoid to roughly the middle of the cell. Those are labeled R, R standing for rhoptries, spelled with an H, rhoptries. The micronemes and the rhoptries help in connecting to the host and secrete proteins that allow the parasite to attach. And then behind that, in the posterior half of the cell, labeled N, you can see the nucleus and the Golgi body. The whole thing, that cell is located in a vacuole inside its host. That's labeled VN in this picture. I think it's usually called a parasitophorous vacuole. That was fun to say. And then Toxoplasma also has this apicoplast. This is what it looks like in cross-section. And chloroplasts in plant and algal cells are surrounded by a double membrane layer. I think we mentioned that in the very first lecture. The apicoplast is surrounded not by 2, but by 4 layers of membrane. And we think that this used to be, way, way back, a eukaryote algae that was engulfed and lost its own nucleus. So the 4 layers of membrane are the layer of the bacterial symbiont, the layer that surrounds it from the host, the layer that used to be part of the eukaryotic algal outer layer, and the layer that used to be a part of whatever engulfed the eukaryotic alga. We actually see this in dinoflagellates as well. Many dinoflagellates are photosynthetic, but what we think they did is engulf a green alga and basically turn it into a symbiont. So we have a dinoflagellate with a green alga inside, which has a blue-green bacterium inside. We have, 3 levels of symbiont here. We've got Russian dolls. I'm not sure if I explained that very well. Maybe I can do a better job in lab. Apicoplasts contain their own genomes. They've got something to do with fatty acid biosynthesis. They also participate in protein trafficking. Proteins are made on ribosomes on the endoplasmic reticulum. You might remember that. Many of those proteins end up going to the Golgi, right? And from there, they might get modified post-translationally and end up getting secreted. I'm assuming you remember this, right? Golgi, things like that. But there's other proteins that end up going to the apicoplast. They get trafficked through there, in particular a protein class called ACP. More about this I do not know. I am not a cell biologist, but I do know that because the hosts don't have an apicoplast, right? If you've got malaria, you don't have apicoplast in your cells, but the malaria parasite does. Which means that if we can work out what the apicoplast does, we've got a chance at designing drugs that will hit the apicoplast and kill the parasite while leaving the host cells completely alone. I'm not up on the latest research in that field, but this is at least a promising strategy for drug development. And it's generally a good strategy is to find some kind of biochemical function that the parasite or pathogen has that the host cell doesn't, and target that with your drugs. Just like penicillin targets bacterial cell wall formation, that makes it pretty safe because your cells don't have cell walls. Your gut bacteria do, which is why you might get a little sick when you take antibiotics. Okay, we can subdivide apicomplexans into conoids and aconoids. Conoidacida and aconoidacida. Some of these names are kind of annoying to pronounce. The ones that have that conoid throughout life and the ones that have a conoid only at one stage. So the ones with a conoid throughout life include gregarines and coccidians. The ones that only have a conoid at one stage include pyroplasms, which include these things that cause cattle disease called red water fever, for example, and hemosporidia, which includes the malarias. And we'll start with the gregarines. What makes gregarines different is, first of all, they are monozenous. They only infect one host in the lifespan. The host is always an invertebrate. There are some that do parasitize marine invertebrates, worms and things like that, but they're very diverse as parasites of insects and incidentally of other terrestrial arthropods like millipedes and roly-polies, and I think spiders can get these too. Spiders I'm not sure, but I know millipedes and roly-polies can. The ones that you're seeing all happen to come from dragonflies. But of course we get these in roaches. The other place I'll get these if we're doing a lab that involves looking at gregarines is mealworms. These are beetle larvae that you can buy live in pet stores to feed to your pet lizard or something like that. They are almost always infected with gregarines. So are crickets. Sometimes when Dave DeSword has extra crickets, he lets my class dissect them, and we commonly find gregarines in their guts. So they are found in a very wide range of insect hosts. Many of them don't obviously do any harm to the host. They probably should be considered commensals rather than parasites. I had this idea that I played with that these would actually make a good system for testing anti-malarial drugs. If you can make a drug that's a candidate for stopping malaria, One way to test it might be to feed it to a bunch of insects and see if they still have gregarines or if they manage to get rid of them. I've toyed with that a couple of times. I haven't quite gotten it to work as a lab for my classes, but maybe one of these days somebody ought to pick that up and try it. You won't see these in lab. Gregarines that are called acephalines have a one-part cell. This is a diagram of one of them called monocystis, which is also a really good one to show students, although we're not going to have time because every earthworm I've ever dissected, like 100%, has abundant monocystis in its seminal vesicles. They feed on earthworm sperm. Earthworms usually make far more than they need, so I don't think these normally cause much damage. I think there's some debate as to whether they cause detectable damage to the host or not. So again, we're not quite sure if these are true parasites or commensals. The feeding stage, called the trophozoite, is kind of crescent-shaped and hard to see because it usually is covered in the earthworm sperm that it's feeding on. So down there at the bottom, you can see that thing that looks kind of like a microscopic tribble. That is a monocystis gregarine. That's a worm sperm germ. Right. I'm imagining all of you laughing uproariously. The ones we're going to see in lab are called cephaline gregarines. And these are the ones where the body's divided by a septum. You can see it really clearly in the one on the right. There is an anterior division of the cell called a protomerite and a posterior division called a deuteromerite. I think I've said this, I'll never ask you the meaning of a word in Greek or Latin, but some people find that learning a number of common roots helps them learn the material. Mero means a division. So merite just means a division of the cell. Proto means first and deutero means second. So first division and second division. The protomerite usually bears an anchoring structure called the epimerite, which is the part that burrows deeply into a host gut cell. Gregarines don't penetrate all the way in the way that malarias and coccidians penetrate all the way in. Gregarines, that epimerite, will penetrate into a gut cell. The rest of the parasite will just hang out. And get it? Right. This picture, by the way, was taken in some roaches, and it shows up in your lab handout as well. The stumpy ones are probably genus Blacticola, and kind of running diagonally across the picture, there's a couple of very long, ribbony gregarines that are probably in genus Protomagalensia, and they do tend to be pigmented, although how much pigment varies. And you can see the nuclei in all of these. The nucleus is not pigmented, so it looks white. When they just hanging out in the gut, anchored in the gut wall and feeding, they're called trophonts or trophozoites. You'll see that word again. It's used for a feeding stage where a parasite just sits there and soaks up food and grows and builds up its metabolic resources. Trophane is Greek for to feed, so the trophozoite is the feeding stage. So the trophozoites will anchor in the gut wall and just soak up, probably just soak up digested food. When they're ready to do the nasty, they'll detach from the intestinal wall and form a stage called a gamont. and the gametes will pair up, a process called syzygy, which I was telling some of you is a good word to know if you play Scrabble. So this picture shows you two pairs of gametes in syzygy. These particular ones came from mealworms, not cockroaches. We did live mealworms that year in invertebrate zoology. So that's, yeah, there might be a third kind of down to the lower left, but most of what you're seeing there is two pairs of gametes in syzygy. Once they're in syzygy, they'll ball up together and form a gametocyst, which looks like this. You can see it's a round cyst divided into two. This will sometimes look like a pair of capital letters D facing each other, blunt end to blunt end. Within that gametocyst, the gametes divide into gametes, and they divide by mitosis because most of the stages are haploid. So that trophozoite was haploid, the gametes in syzygy were haploid, and now that they form a cyst, each one divides by mitosis to make gametes. Within the gametes, or within the gametocyst, the gametes pair up and form zygotes, which are diploid. But the zygotes probably divide by meiosis and then by mitosis to form 8 haploid sporozoites. That's the infected stage. So stage 1 is a sporozoite that gets eaten by a new host. In the case of cockroaches, it's because they eat their own poop. So a poop-eating cockroach gets infected with a sporozoite. The sporozoite attaches to the gut, burrows into a cell and hangs out and grows and becomes a trophozoite. That's stage 2. Then at 3, you see 2 pairing up in syzygy. 4 is that gametocyst. 5 is mitosis happening. 6, the gametes are pairing up to make zygotes. And then 7, the zygotes immediately undergo meiosis and then mitosis to make these oocysts, each of which contain 8 haploid sporozoites. And round goes the great circle of life that moves us all. And again, you should have this in your lab handout, and a lot of it you're going to see if you haven't already. Okay, so by the power vested in me by the state of Arkansas, I am going to stop recording right now.